Hello students, good evening all. Audio visual clear?
Yes, sir.
Good evening, sir.
Good evening. Okay, last day in the core paper class, we discussed that the diversity of microbial world can be grouped into cellular organisms as well as acellular organism. Yes or no? Cellular organisms as well as acellular organisms, and under the cellular organisms there will be bacteria. As per the domain model, bacteria could be of two types: Archaebacteria and Eubacteria, as well as fungi, protozoa, and algae. These are under microbial organism in cellular category. But acellular organisms are viruses, viroids, and prions. These all are in the syllabus, unit 2 syllabus. So till now we covered, in last class we covered the Archaebacteria, Eubacteria in details. Fungi, protozoa, algae remain, we will cover in next classes. Fungi, protozoa, and algae. These are discussing lot on description. Today we going to discuss about the acellular organisms, that means viruses, viroids, and prions. So let's start. Acellular organisms. Acellular organisms. Virus, viroids, prions are called acellular. Viroids and prions are acellular organism. Why? Because it is clear that from the name, acellular means there is no proper cell structure. Acellular means no proper cell structure. What structures present inside a cell? Tell me. Inside, outside the cell. What are the structures that you see inside or outside of the cell? A common cell.
Sir, inside the cell we see cell organelles and
Cell
cytoplasm.
organelles. And outside cell wall, cell membrane, right? So these all structures, that means cell wall, cell membrane, inside cell organelles, cytoplasm, all structures are absent in case of acellular organism. That means virus, viroids, prions, they are acellular because properly cell structure absent means which structures are absent? Cell wall absent, which is the integral property of the cell that creates the protection layer outside cell wall. Cell membrane is very important part of a cell. It's basically used for the transport purpose, transport of nutrients. Cell membrane selectively transport some molecules, nutrients, etc. Cell membrane. So they all lack cell wall, cell membrane, as well as organelles, cell organelles. That is why they are all acellular organisms. And we could not classify these organisms into five kingdom model or three domain model. Can we classify these organisms? Can we classify these organisms into kingdom model or domain system? What is the criteria of kingdom model classification and domain model? It's very clear in the domain model there should be a ribosome or rRNA present. Yes or no? In this case, virus, plants, no organelles, that means no ribosomes are present, so we cannot classify this organism inside the domain model as well as kingdom model also, because kingdom model, the Whittaker kingdom system, the features that Whittaker considered, cell wall, cell membrane, nutrition, mode of nutrition, etc. etc. Correct. So we cannot consider these organisms, viroids, and prions into the kingdom model as well as domain model. So these organisms left aside into the acellular organisms. We cannot classify into the any classification model because of acellular property. Any classification that we previously discussed, that is three kingdom, four kingdom, five kingdom, well accepted five kingdom, and after that it is modified into domain model. So no one classification system can classify viroids, and prions. Clear? Now let's discuss more about the virus, specifically the group virus. Start in more details. First of all, we look for the discovery of viruses. Which scientists are majorly play a role in discovery? Some important scientists started with Louis Pasteur. The starting scientist, Louis Pasteur. Yes, Louis Pasteur has role in the virus discovery because Louis Pasteur worked on rabies virus, work on the disease rabies. Yes, he did not know about the causative agent, it is a virus or not, but he worked on rabies disease and he knew that this rabies disease is easily transmitted from one host to another host. Clear. So we cannot. We should consider Louis Pasteur contribution inside the virus discovery. That is, Louis Pasteur during his studies of rabies disease, he knew that this disease is transmitted from one host to another. This is transmittable from one host to another host, healthy host. At that time, Louis Pasteur did not know about the causative agent, about the causative agent of rabies disease, but we cannot overlook this historical contribution of Louis Pasteur. Next scientist after the Louis Pasteur work, it is Ivanovsky. Louis Pasteur, this work is in 1884. Ivanovsky showed the work in 1892. After 8 years, Ivanovsky showed that he worked on tobacco mosaic disease, that is a plant disease. And he showed that with simple experiment, the causative agent of tobacco mosaic disease is smaller than bacteria. Smaller than bacteria. Clearly, he also do not know about the causative agent, which is the virus, but he clearly knew that the causative agent structure is smaller than bacteria. The average size is smaller than bacteria, and it is proved by simple experiment. And we will conclude this experiment in short way. Experiment of Ivanovsky. Experiment of Ivanovsky. Very easy and simple experiment. He produced extract. Tobacco mosaic disease actually affected in the tobacco leaves and mainly caused the mosaic pattern. He produced the extract of disease leaves of tobacco. So clearly, he take some affected leaves and produce the extract. Clear? Now, this extract is passed through the porcelain filter. Porcelain filter, and the unique about the porcelain filter is pore size is smaller than bacteria. Porcelain filter pore size is smaller than bacteria. That means bacteria cannot pass through the porcelain filter. Yes or no? Porcelain filter pore size is very small, and it is very small and smaller than the bacteria also. That's why if you want to pass the bacteria solution, bacteria cannot pass. Correct? Online students. So after the extract is passed, after filtering, two things are produced. After filtering the materials, this extract, two things are produced. Porcelain filter, one is solid and one is liquid. That means after the passing through the porcelain filter, some materials is passed through and some materials is retained. Clear? The materials that pass through the porcelain filter called filtrate. Filtrate means those materials are passed with the porcelain filter, within the porcelain filter, that is called filtrate. And after filtering the next portion which is not retained inside the filter, it is called retentate or residue. It's called residue. So after filtering the extract, two things are produced. One is solid portion residue that is not passed, and the passed portion through the porcelain filter is called filtrate. And he observed that the filtered portion is the infectious part. Infectious. The residue portion do not have any capacity of infection. So what we conclude after the experiment? Can we conclude the retented or filtered portion contain the causative agent? And if the causative agent remain in the filtrate, that means it should be smaller than bacteria. Yes or no? So what will be the
Yes.
conclusion? What will be the conclusion after this simple experiment of Ivanovsky? We can easily conclude now the filtered portion has the infectious property and the filtered portion only produced when the porcelain when the molecule pass through the porcelain filter, and the molecule should be the material should be lesser than the size lesser than the bacteria. हाँ या ना ठीक तो so conclusion from this experiment Ivanovsky clearly conclude that tobacco mosaic disease, the causative agent is smaller than bacteria that we written here. Tobacco mosaic disease causative agent smaller than bacteria. This experiment further uh, considered and further proved by Behring. Next scientist, scientist number 3, he also do same experiment and the result is same. So scientist number 3, after Ivanovsky experiment, M.W. Behring, 1898. He do all experiment at 1898. So confirmed this above experiment further repeating, and he named the filtrate which is the famous code Contagium vivum fluidum. It is a Latin sentence. Its mean, the English meaning is infectious fluid. This filtrate, this filtrate M.W. Behring called Contagium vivum fluidum, and it means infectious fluid or virus. And actually the virus mean poison or toxic materials in Latin. So who coined the term virus? Who coined the term contagium vivum fluidum? Actually, Behring. After confirming the Ivanovsky experiment, same experiment, repeating the same experiment and confirm that the causative agent is smaller than bacteria and we should name in different way, not the bacteria. So the name will be given by virus or contagium vivum fluidum. After Behring experiment, Stanley in 1935 he crystallized the tobacco mosaic virus. So virus crystallization is possible, and that is observed and done by Stanley. So that is basic history about discovery and some scientist contribution. And number five scientist, you all know that Hershey Chase. You hear the name all Hershey and Chase. In their beautiful experiment, radioactive experiment, what they show about the virus? Hershey Chase experiment, what they conclude? Tell me, online students, Hershey Chase experiment, what they conclude after this experiment? And this experiment is conducted in 1952. ক্লাস 12 সব পড়ে এসছো তো? Conclusion of Hershey Chase experiment
Sir, the DNA is the genetic material.
of which virus? All virus or particularly one group of virus? Of bacteriophage, not all. So Hershey and Chase studied with T2 bacteriophage. T2 means temperate bacteriophage. T2 bacteriophage, and after this experiment, they conclude that DNA is the genetic material of bacteriophage T2. So now name some notable contribution of virus discovery history scientist. And next we look for the properties or features of viruses. Features of viruses. Features number one, viruses are ultra microscopic. Ultra microscopic. What does it mean, ultra microscopic? Microscopic, we all hear the term microscopic, that means that we see inside the under the microscope. So what about the name or the word ultra microscopic? Electron microscopy, that means the structure which is not observed under normal microscope, it should be observed under electron microscope. So viruses structure cannot obtain in normal microscope, regular microscope, laboratory based microscope, bright field microscope. Clear? Have to be done observed under the electron microscope. That's why the ultra word is came in prefix, because ultra microscope means it needs powerful microscope that zoom the structure in multiple way, many way. That microscope is ultra microscope, and we know that the microscope needs for the virus discovery or virus observation is electron microscope. That's why the virus structure is ultra microscope. So it only visualize by electron microscope. That's why virus structures are ultra microscopic. All virus, not single one. You take any name of virus and it will be ultra microscopic. Point number two, features number two, they are obligate intracellular parasite. Obligate intracellular parasite. Now let's expand this term. Obligate intracellular. Obligate what mean? Obligate. Obligate mean আমরা পেয়েছি এর আগে ওয়ার্ড obligate anaerobes পেয়েছি obligate mean absolutely necessary, without that they cannot survive. That time we can use the word obligate. Clear? Intracellular. Intracellular means inside the cell. Cellular means cells, intra means inside. So they should be present inside the cell. And parasite means they take all the metabolism and nutrient related work from the host. That is why it called parasite. So it absorb or reproduce, better say, grow and reproduce inside host. So now combine together and form the sentence. Obligate intracellular parasite means it should need a healthy host for their survival. Yes or no? It should need healthy host. Without that, they cannot grow, they cannot reproduce. Healthy host for their survival. That is why the words are given here in the feature. Obligate, should need, intracellular, a particular host cell is needed. Inside the host cell, they grow and reproduce, and it is for the survival process. Features number two clear. Obligate intracellular parasite. Answer. Features number two clear to all.
Yes,
Yes.
sir.
Number three feature. Host specific. Viruses are host specific. What does it mean? It means that it needs particular host. A virus, particular virus prefer to choose particular host for their growth and reproduction. For growth and reproduction. Give an example of host specificity of virus. Give an example, student. Give an example of host specificity. The meaning is very clear. It needs particular virus needs particular host. Give an example of that feature. Try to answer.
টোবাকো মোজাইক ভাইরাস যেটা
হ্যাঁ, ওর হোস্টটা কি?
টোবাকো লিভস
Tobacco leaves or tobacco plant. So only infect in the tobacco plant, not in mango or other plants. Yes or no? Likewise, bacteriophage only infecting bacteria. ব্যাকটেরিওফেজ কি হিউম্যানকে ইনফেক্ট করে? Can bacteriophage infect in human cell? No. Why? Because particular virus needs particular host for their growth, for their metabolism purpose work, for their reproduction. So good example, any example you can give it. Bacteriophage only grow and reproduce inside the bacteria. That's called host specificity. Bacteriophage cannot grow in other cellular organisms like fungi, parasites, that means protozoa, algae, any cell. Other cell they cannot grow, but inside the bacteria they need grow and reproduction work for survival bacteria. That's why it's called host specific. No metabolic activity. Clearly, they are parasite. We previously written here, it is parasite. That means they have no metabolic activity. They have no metabolic enzymes for their growth. They need some enzymes help from host. So they prefer to grow host because they need metabolic activity. They need host for metabolic activity. They cannot do it by its own. Feature number 5, easily transmittable. Easily transmittable means it can spread easily from one person to another person, one host to another host easily, easy way. So rapid transmittable process they have. One host to another. What is the necessary of transmission? Transmission of viruses? Survive. What is the necessity? Because survive, because they need healthy host. All the time they need healthy host. Yes or no? All the time they need healthy host for their survival. So always virus look for healthy host. After entering a healthy host, they manipulate and grow inside the host, and the host will be dead after some time. So virus next target is another healthy host. That's why the transmissible process is very important in virus life cycle. This transmittable process is very important, and transmit can be done by air through, water through, food, etc. And it is very swift process, easy, rapid process than other cellular organism. That means virus can spread easily. We can easily conclude. Yes or no? From other organism, virus are spread easily and rapidly. And we also look in COVID situation that it spread havoc in human and quick spreading হচ্ছিল. right? Because virus a character ওটাই, virus need healthy host, and for that, for the survival, they need healthy humans or healthy host for growth. That's why transmission or transmittable process is very important in their life cycle of any virus. Number 6, easily crystallizable. Yes, that features is shown by Stanley, that virus can be crystallized. Virus can be crystallized because virus contain large amount of protein in their structure. When a structure is crystallized structure, the structure is formed crystallized in biological sample when proteins are in huge concentration. And from that view, it is clear that virus is crystallized. From that view, it is clear that virus contain good amount of protein in their structure. All features are clear. Virus features or properties in general scenario, not particular virus. In all virus aspect, some important features or properties. Properties are clear.
Yes, sir.
So question. So question can be asked from the features of virus in tricky way, like that viruses have intermediate between living and non-living. Explain or justify. How can you explain that question? Viruses are intermediate between living and non-living.
ভাইরাস যখন হোস্ট সেলের মধ্যে থাকছে তখন সে লিভিং প্রপার্টিজ শো করছে, যখন ভাইরাস না হোস্ট সেল পাচ্ছে না তখন সে নন লিভিং প্রপার্টিজ শো করছে। আমি এটা একদম ইজিলি ভাবে বললাম।
আচ্ছা,
জীব জ্বরের মাঝামাঝি অবস্থা।
হ্যাঁ, তো 3, if the marks distribution is 3, so one property is not enough. Minimum 3 to 4 property should be written.
সেক্ষেত্রে কি ভাইরাসের ফিচার লেখা,
এই তো ফিচার গুলোর মধ্যে থেকে তোমাকে তো আইডিয়া করে বার করতে হবে। So তোমাকে যেটা করতে হবে, the statement is correct or wrong? Statement is correct. Viruses is intermediate between living and non-living property. So living like features তোমাকে add করতে হবে. What are the living like features of virus? And second one, you should write over non-living features of viruses. Now take some living like features and give some non-living like features. Mention.
ট্রান্সমিসিবল,
Transmission. Living like features number 2.
হোস্ট স্পেসিফিক,
মেটাবলিক অ্যাক্টিভিটিস তো দেওয়া যায়।
আরেকবার বলবে?
বলছি মেটাবলিক অ্যাক্টিভিটিস তো দেওয়া যায়।
Yes, yes. Metabolic activity inside the cell.
স্যার ওটা তো নন লিভিং এর আন্ডারে আসছে কেন কি নো মেটাবলিক অ্যাক্টিভিটি অফ देयर ওন?
হ্যাঁ, but metabolic activity inside the host, that is the living property. Outside it has no metabolic capacity, it is the non-living property, but inside the host cell it has the metabolic property. by borrowing the host mechanism. So it is the living like property. Inside the cell লিখতে হবে, inside the host cell. Number 3, growth and reproduction inside the host cell, that is also another living like property. Now non-living properties. First of all, it is crystallized. So crystallized structure only done in non-living things. So virus can be crystallized. So it is under the non-living properties of the virus. Number 2, outside the host cell, they are non-living like properties. They cannot grow, or they do not have any metabolic properties. No metabolic property outside the cell. Number 3, only grow inside the cell, or cannot grow, reproduce, better, cannot reproduce outside the host. And live like inert material and present like inert material. That means non-animate, inanimate object. So these are some features of non-living property. So answer of the questions like that, the statement is correct. Okay. And now give some properties of living features of viruses and give some properties of non-living features of viruses. And answer complete. ঠিক আছে? তো কিভাবে আনসার লিখতে হয় অলরেডি বারবার বলছি, কোশ্চেন যা আমি বলবো সেগুলোকে নিজেরা লিখে আমাকে চেক করাবে, না হলে বুঝবে না কিভাবে লিখতে হয় আনসার। ওকে, so features of viruses are done. Next, the structure. Next we look for structure of viruses. General structure. Virus structures composed of nucleoprotein. Nucleoprotein structure mean it composed of two materials. Break down this word into nucleo and protein. From nucleo it will be RNA and or genetic material, better to say RNA or DNA, genetic material, and the protein means another material, that is biomolecules protein. So two composition structure inside the virus structure. One is nucleic acid. From the nucleo we can say it is composed of nucleic acid, and other is protein. And from the crystallization part, Stanley observed that the virus portion of the protein is much more high than the nucleic acid. This protein portion is generally inside the viruses are higher portion, contain higher portion of protein and lesser portion of nucleic acid. The protein portion of the virus are also known as capsid. This protein portion form outside structure. So protein, where the protein is lies inside the in the viruses in the outside structure, and this structure is called capsid. This structure is called capsid, and the capsid structure, these proteins are accumulated each other. That means not a single protein make the structure; it is multiple of proteins, and the unique structure of proteins that make up the capsid called capsomere. Called capsomere. So capsid is not a single protein structure; it is multiple of various protein, many protein structure, and the unique structure of the capsid is called capsomere. Capsomere. So capsomere is the unique structure, and the whole structure, large structure that form the outside material is called capsid, polymeric structure. And capsid has several symmetry. Capsid symmetry. Capsid symmetry means the arrangement of capsomere and make the geometrical shape of outside structure. What type of geometrical shape it produce after the capsomere arrangement is called capsid symmetry. তার মানে outside structureটা কি রকম দেখতে? Okay, so capsid symmetry can be of generally two types. One is helical type, helical symmetry. So capsomeres are arranged together to form helical shape of capsid. It is very common. Another very common capsid symmetry is polyhedral. Polyhedral type. Polyhedral symmetry. Helical symmetry looks like capsomeres are joined and form helical shape like that. These small circles are called, each are capsomere, and whole structure is capsid, and it is helical shape. So helical symmetry. And other more common symmetry is polyhedral. This type of shape is called polyhedral. So outside structure totally made up by the protein is very clear, and the arrangement can be different, and more common two arrangements are helical and polyhedral. This helical type of symmetry observed in TMV virus, good example is tobacco mosaic virus, are helical type of symmetry it show. Polyhedral type of virus, good example bacteriophage head, poliovirus, all are polyhedral. So the higher proportion protein, how does it make the structure of virus? It completed. Next is the nucleic acid. So other composition of the virus is the nucleic acid, means genetic material of the virus. Nucleic acid is the genetic material of the virus. Nucleic acid can be of different types in case of viruses. It can be DNA, or it can be RNA. So RNA is the genetic material only found in the virus. Yes or no?
Yes.
Other than viruses, all other living animals, living systems, living organisms contain DNA in their structure, and also bacteria. No bacteria contain RNA in their genetic material. All bacteria contain DNA, even archaebacteria also. Yes or no? But only some viruses have RNA in their genetic material. So, based on RNA nucleic acid type, viruses can be different types. So, nucleic acid is various type possible in case of viruses, DNA or RNA, and even DNA also variant in virus. Some virus contain double strand DNA. dsDNA means double strand DNA. Some virus contain single strand DNA. So single strand DNA also another unique features in case of virus. Most of the organism contain double strand DNA, that means Watson Crick pairing of double strand DNA helix. But some virus also contains single strand DNA. Another uniqueness about genetic material of virus.
Virus can contain DNA and also RNA.
RNA, even DNA can be of single strand type. That is another uniqueness. And RNA, here also some different type. Some virus contain single strand RNA and many other contain double strand RNA. Now look at that. Double strand RNA, another unique features. Normally RNA remain in single strand form, but in some case RNA virus contain double strand RNA also. Clear? So variety of types of nucleic acids can be found in virus. Online students, clear?
হ্যাঁ স্যার
Yes, sir.
এখানে আবার টাইপ থাকে সিঙ্গল স্ট্র্যান্ড আরএনএ ক্যান বি অফ ফার্দার সাবক্লাসিফাইড ইনটু টু টাইপস ওয়ান ইজ প্লাস এসএস আরএনএ other is minus ss RNA. Positive strand single strand RNA, positive and negative single strand RNA. Another different types. Clear? Why it is called positive, negative? It is different segment. It is in the virology semester, one virology topic. We not here conclude or discuss here. Just mention single strand RNA can be of further two types, positive and negative. So virus structure overall basic idea clear. What makes a virus structure composition? It is protein and nucleic acid. Protein portion is higher, nucleic acid portion is lesser, but variety of types. DNA or RNA, yes. Another mentioning this, either containing DNA or RNA. No virus contain both DNA RNA as a genetic material. এরকম তুমি পাবে না। That means একটা ভাইরাসের মধ্যে DNA RNA দুটোই genetic material কাজ করছে। This virus is absent in nature. Only contain either DNA or RNA, not both act as a genetic material. Clear? RNA virus do not contain DNA in their genetic material. DNA virus do not contain RNA as a genetic material. So it is DNA virus, it is RNA virus, either or, not both. So what is the function of protein, means capsid, and the nucleic acid? Capsid generally function as a outside protective layer in virus structure. So capsid function is protective layer. Protective layer. Protective layer of nucleic acid. It protects nucleic acid. That means capsid is present because of nucleic acid protection. Yes or no? Clear? And nucleic acid do all the things. The function of nucleic acid is it is used as a growth, reproduction, etc. etc. So nucleic acid is the infectious material of viruses. infectious material. That means nucleic acid is the main things that will inside the host cell cause growth reproduction of viruses, not the capsid part. Capsid is basically for protection of this nucleic acid portion.
WBC থেকে ক্যাপসিড রক্ত করে।
হ্যাঁ, মানে নিউক্লিক অ্যাসিড কে প্রোটেকশন দিচ্ছে, বিকজ উইদাউট নিউক্লিক অ্যাসিড ভাইরাস ইজ নাথিং। Understood? Point clear? Structure and function of two portion.
হ্যাঁ স্যার।
So virus overall properties, basic structure done, and next acellular organism list here is viroid. Viroids discovered very recently, 1971, by T.O. Dinner. Discovered by (1971) T.O. Dinner. And he observed that viroids are free RNA and lack of capsid. Viroids are free RNA. That means its structure only contain RNA. Do not contain capsid. Clear? No capsid protein layers are present inside the viroids. And the RNA here is specially formed, circular form, single strand form, low molecular weight, that means only contain 250, the length of the RNA 250 to 400 nucleotide long, small size. And another features of viroid RNA do not contain any protein. Do not code protein. Normally, the function of RNA, mRNA is form protein. After the central dogma process, after RNA formation, it will enter into the protein formation, that means translation step. But this RNA, the viroid RNA, do not have any codon. No protein is formed. So some features of viroid RNA: circular, single strand, low molecular weight, and number 4 features, do not contain any codon. That means enter into the protein formation translation process. So viroid structure is smaller than the virus because do not contain any capsid layer outside. So it is smaller than the virus. Viroids are smaller in size, smaller than virus, and viroids also can infect host cell. That's why viroids are called smallest living infectious agent. smallest known agent of infectious disease. And the next question is, how does viroids cause infection? Simple, describe, বেশি করব না, এই লেভেলে বোঝা পসিবল নয় অনেক কিছুই, বাট mechanism of virus infectious property, it is done by RNA silencing. It is done by infectious is done by RNA silencing. RNA silencing is very important phenomenon occur inside the eukaryotic cell. RNA silencing, just basic meaning, the viroids is the RNA, virus structure contain RNA, and viroids RNA interfere with normal cellular RNA, and this cellular RNA either blocked or cannot enter into the translation process. Clear? তার মানে যদি সেলুলার আরএনএ এন্টার না করে ট্রান্সলেশনে, it makes problem inside the host. Proteins hampered, proteins production hampered, ultimately host cell will be died. Clear? So that is the silencing process, RNA silencing process. This viroid RNA silence the cellular RNA.
ভাইরাসের তো কোনো প্রোটেক্টিভ লেয়ার থাকে না ক্যাপসিডের মতো, ডাইরেক্ট নিউক্লিক অ্যাসিড নিয়ে যখন
direct nucleic acid নিয়ে ইনফেকশন করছে, and direct this nucleic acid is highly resistant. That's what the special structure is circular. So structure is very resistant than the RNA virus. The RNA of viroid is very resistant, highly resistant than the RNA of virus. Normal virus RNA is susceptible to any kinds of RNAs, enzymes treatment, but this RNA, viroid RNA, is resistant. That's why it can survive, and when it enter into the eukaryotic cell, it causes this process, RNA silencing. RNA silencing means this viroid RNA silence this cellular RNA and interfere to enter translation process. It's very basic RNA silencing meaning, but details obviously এখানে ডিসকাস করার মতো স্কোপ নেই. It will be in the further molecular biology sectioning, RNA silencing. What is RNA silencing and how does it occur? What are the complexes are involved in RNA silencing, etc.
আমরা বায়োলজিতে করব না।
না, এটা molecular biology part. Clear viroids? And next and last, acellular organism, prions. Coined the term prions by Stanley Prushiner. (1862). Prions are actually abnormally folded infectious protein without nucleic acid. So here it is in different scenario. Here no nucleic acid is present. The protein part is infectious. And this protein is not normal. It is abnormally folded. Abnormally folded means every protein has their own structure. It can adopt primary structure, secondary structure, or tertiary structure. Protein has different level of structure, primary, secondary, and tertiary. Majority of the protein functional in the tertiary structure. Clear? And when this tertiary structure is folded or abnormally, that time the problems form. That time the protein. So when the normal protein the adopt different type of structure, tertiary structure, that time it is called abnormally folded protein, and this protein can be infectious. Without nucleic acid, these proteins can be infectious and cause diseases. Clear? Point clear? Online students. So prion cause many neurological disorder, neurodegenerative disorder. In human and other animals. So prions basically cause one type of disease, that is only neurodegenerative disease, means neurological symptoms or disorders. All symptoms are neurological disease. Clear? And some examples of prions related disease: bovine spongiform encephalopathy, short form BAC, and common name is mad cow disease. Common name is mad cow disease. We previously said that it is mainly prion cause neurological problem symptoms, and mad cow disease another. It is actually neurological symptom, neurodegenerative disease. BSE is the actual name, bovine spongiform encephalopathy, BSE, or mad cow disease. And same looks like disease occur in human also, and called CR Jacob syndrome. Short form CJD. Similar like symptoms occur, mad like symptom, neurological problem, and it is caused by prions, not any other organism, not bacteria, not virus. It is prion related disease. Another good example, scrappy. It occur in sheep. Clear? Next similar question, how does prion cause infection? Mechanism. Simple way. General mechanism. It is better understood. Prions, how does it occur? It can transmit or cause disease. It is better understood in scrappy diseases. scrappy diseases that occur in sheep. Here, the prion protein called PP, simple PP, or actually it is called, in case of scrappy, the prion protein is named as PRPSC. It is terminology is PRPSC. That means prion protein of scrappy, PRPSC. Prion proteins it first interact with normal protein. That means normally folded protein produced inside the cell, but when prions is present, these interact with the normal protein. And normal protein is called NP. And in case of scrappy, the name of normal protein is PRPC. PRPC means normal cellular protein. Cellular protein. C for cellular. PRPC. So how does it infect? First of all, PP interact with the NP, and now NP is abnormally folded. After that, after the interaction, what happened? This NP means normally folded protein become PP. NP convert into PP. So majority NP are now convert into PP, and PP now oligomerize. That means prion proteins start to interact each other and form oligomerize. Oligomerize means clump formation done. So after NP convert into the PP, after this interaction, the PP now oligomerize and cause problem. And cause disease. So that is the normal simple mechanism of prions related disease. Better understood in the scrappy disease, scrappy prion protein, exact name PRPSC, normal protein is PRPCC for cellular, SC means scrappy protein. After the interaction, normal protein convert into the prion protein, and now prion proteins are oligomerized and some causes the disease symptoms. Start the symptoms. So এটাই আমাদের বিলিভ করা মানে প্রবলেম মানে বিলিভ করতে পারি না কিভাবে one protein can cause infection. How a single protein cause infection? By that way. It cause or trigger the changes of normal protein inside the cell. And normal protein, when it convert, it become oligomerized and start to store inside the cell. It is not এটা মানে ক্লিপ্ড হয় না ও বেরোয়ও না ওভাবে স্টোর হয়ে থাকে and cause problems and mainly cause problem. Understood the prions mechanism of infection. Clear? Viroids example দেওয়া হয়েছিল previously. Viroids example. I think I missed it. Viroids related disease. Example. Potato spindle tuber viroid, PSTV. Good example. Potato spindle tuber viroids cause potato spindle disease. Viroids লিখবে কিন্তু V for year virus, not virus. Another example of viroid disease, coconut cadang disease. So from the example, it is clear that viroids cause plant-related problem. Look at that example. And the prion cause animals human related problem, because neurological means only present in animals and humans, mammals. So neurological problems disorder created by prions, and plants related problems majorly created by viroids. Clear? Acellular organism. All students answer. Acellular organism, virus, viroids, prions. Clear overview?
Yes, sir.
So make a comparison.
Viroid causative diseases second point কি লেখা?
Coconut cadang disease, cadang disease. কোকোনাটে হয়, কোকোনাট ট্রিতে প্রবলেমটা। So make a comparison between all three acellular organism, and that will be much more clear. The concept will be much more clear if the comparison is done. Features, virus, viroids, prions. Feature number 1, nucleic acid. Present or absent? Answer me.
Present.
In case of virus, nucleic acid present. Viroids
Present.
present. Okay, এখানে আমরা better mention করব DNA. Nucleic acid can be DNA or RNA, but viroids only RNA. No DNA is there. Prions, nucleic acid absent. Features number 2, presence of capsid or protein layer. Now answer me.
Present. Absent. Absent.
Virus present, capsid present, viroids absent, prions. Obviously, protein is the infectious part, so protein abnormally folded. You can written over here. It is abnormally folded. Viewed by. That means how can you visualize these structures, acellular structures? Viewed by viruses, viewed by
Electron microscope.
electron microscope. Viroids,
Crystallization.
crystallization or sequencing, better, not crystallization, it is sequencing, nucleotide sequencing. Crystallize only done when proteins are in high concentration. Here is no protein, so no crystallizable, no question of crystallization. So here it will be viewed by nucleic acid sequencing. And prions only visualize when host cell are damaged. Other than you cannot observe it is prion because it is a normal protein. When host cells start to damage, then you can visualize that these host cell can contain prion. Clear? সো আগে থেকে তুমি বুঝতেই পারবে না ওর মধ্যে প্রাণ আছে কি নেই। Next important features, number 1, 2, 3, 4, affected by heat and protein denaturant. Virus will be affected or not in heat? Affected? Yes or no? Online students answer করছই না। Virus affected by heat or protein denaturing agent? Yes or no? Am I audible? Clear? Audio clear? Online students, am I audible? Audio clear? Affected by heat and protein denaturant virus? Yes, affected. Inside viroids কি হবে? Prions,
নিজেই তো প্রোটিন ধরনের
protein, but it is highly resistant, cannot affected by heat and protein denaturant. Negative. Affected by radiation or nuclease enzyme. Nuclease enzyme actually cleave the DNA RNA portion, and radiation also affecting DNA and RNA. So viruses can be affected or not? Virus affected? Yes. Viroids, RNA present, radiation nucleus can affect, but prion?
no.
Number 6, host cell required to grow and reproduction. Virus,
Yes.
yes, and any kind of cell, any cell. It can be bacteria, animal cell, plant cell. In all type of cell they can grow. Yes or no?
Yes.
Viroids specifically grow inside the
Plants.
plant cell. And prions mainly animal cell and human. So that is the comparison chart between 3 acellular organisms in our list: virus, viroids, and prions. If the features are clear, now you can understand what is the concept of virus, viroids, and prions. Clear? So in next class, we are going to discuss about the virus in more details. That means viruses life cycle. That is not covered here. Here we will cover the discovery part, structure part, and just general idea. But the life cycle is still not covered. So life cycle of viruses will be in the next class, and specifically we discuss about the life cycle of simple virus bacteriophage life cycle. Bacteriophage life cycle.